of the administration, which many of us find deeply frustrating because it's not actually what we were ever very good at in the first place. And that's an extremely difficult tension, but it is a very typical tension that I see everywhere, not only in India.
Okay, we have time for one last question.
I think the magic word you just said, research overheads. What makes institutions care about the fact their PIs get the grants? If the overheads are sufficient, as they are, for example, in NIH foreign grants, institutions want to build the ecosystem, invest in them, so they have more money. Otherwise, it's not uncommon for the leadership of institutes to sometimes see small grants as a liability to the institute. When I speak from a very personal perspective here, when somebody brings in a 25-30 lakh grant, the first thought going through my mind is, that's actually a drain on my resources as opposed to adding anything. Because it barely brings in any institutional overhead. It builds more stress upon the system. And I really think that in the Indian ecosystem, we need to provide a bit more overhead than we currently do. And you know that even that 5% that's there gets capped off after you hit 1 lakh or 2 lakh. So you have typically these 2 crore grants with 2 lakhs overhead, which at CSR we're able to manage a bit because we have internal resources. But when I think of other institutions, this problem is really serious. And I think you already pointed out, it's about overheads.
Yes.
Can I respond to that? I really think the scientific community should push the Indian science leadership a bit more. I see it becoming more and more disengaged. If you look at yesterday's panel, or the day before yesterday's panel discussion, and you look at the history of India Alliance, there was a lot of push from the scientific community to set up the India Alliance. And I see the scientific community now disengaging from this dialogue with the science leadership. And I think you should engage the science leadership a bit more, whether it's the minister, whether it's the secretaries, and try to get these structures in place. And I think it is a joint responsibility between the science community and the science management people to reform our structure. And you really should push a bit more. You should be more demanding of our leadership.
Thank you, Shailja. Maybe we'll just take that question from Deepak as well.
Can I go?
Sorry, Deepak has a question, yes.
Yeah.
Actually,
last question.
So.
Okay, yes.
Yeah. Actually, since I understand that the United Diet Stand is a theme of the discussion now, that we need to unite together and we need to foster the more interpersonal or inter-lab collaboration for enhancing the research funding possibility and its smoothness in striving the project for solving national level problems. So, having experienced very well in Germany, I would refer to this DFG, German Research Foundation, as a best example for this. They have a very strong mechanism in place called SFB, Spatial Forschungsbereich, and that is called German Research Group. So where they have official program where the PI number of team of 10 or 15 PIs come together, contribute A to Z issue, solve the major problem, and they all get funding at the same level, same time. So I think such mechanism, if we can introduce in India, that will solve many problems, and it will also solve the issue of copyrights and the shareholding of in the idea being proposed or being addressed. So I think this is a very important mechanism which we should introduce.
Okay. A plea for you. Find your own solutions that work best for you. Look at what everyone else in the world does, but don't copy any of them. I know the DFG. I'm working with the DFG on a different program because they perceive that they have weaknesses in their system and they want to change them by moving them to more EU cases in another part of their agenda. Go and cherry pick the best things you find in everyone else's system and form your own system that works best for you in India.
Thank you very much. So I think on that note, we'll sort of bring this session to a close. Are there any final comments that our panelists would like to make? Jitu, do you have any?
No, I just think the two elements of reform. One, One in being able to create sustainable opportunities for, for research management. And the other is having, having the younger PIs literally reach out across institutions and form communities that will feed back exactly the kind of things that Shailaja was talking about.
Thank you. Ashok?
I don't have anything to add to what I've said.
I think I said enough.
I think we're done.
We're done. So please join me in thanking the panelists. Thank you for your time.
Thank you, Mike. Thank you, Shailja, Ashok, Jitu, and Savita for a very interesting discussion. And I'm sure the audience will have more chance to discuss with the panelists during the breaks. So in the interest of time, we will have a 20-minute break so that we can cover the remaining talks today within the schedule. So I request all of you to be back within 20 minutes after the short break.
for a very successful 10 years and the science rendering and giving all the support for another decade. And as fellows, we think that we have a role to play here, and we will definitely want to see that you have another successful decade, and that other fellows will be here to say the same similar thing in the next 10 years. I also just want to say I'm here with my team who have supported me, who have always been with me for almost 2 years into my fellowship. And so I have Sandra and Suraj here and Lydia here, who would be talking at some point during my presentation. So I'm going to try to move away from the biomedical sciences a bit, which is always a good thing for some of us, because then we can get to talk about some other things, some other interesting behavioral issues that affect most of us. And most importantly, I'm going to try to talk about the issue of drinking problems in young people and adolescents, and try to argue and present a case for coming up with innovative strategies. I mean, as scientists, I think what we are encouraged and motivated by is to come out with innovative strategies in whatever we do in our labs. And there's a very good reason for that, because obviously we have to keep innovating, given the way trends are beginning to be recorded. We have a problem, and we think that we couldn't continue doing, what some people have said in the past, work as usual. We have to start thinking about newer ways of doing things. And so that is my passion, and my passion is to try to come out with newer ways of addressing the treatment gap for adolescent drinking problems using digital health technology. I'll just start off by giving you a brief of what the current situation is and why we should be interested in this topic. Globally, 5% of all deaths, the WHO reports 5% of all deaths in 2016 were attributed to alcohol directly or alcohol-related problems. And if you juxtapose that with the other metric that we use to calculate the DALYs, which is years lives lost with disability, you would realize that there is a certain increase in burden. And it has been increasing from what this slide shows you. It has been increasing between men and women. It has been increasing from 2004 to 2012. We have seen almost threefold increase. And in 2012, what we are seeing is that it does appear that it's stagnating with men, but what we are seeing is that for women, it is increasing. And that's, for me, it's a worrisome trend. And if we may begin to think of why this is the case, and given that we already know about half of mental health disorders or mental health problems have their onset in childhood and adolescence, we want to begin to think about why this is the case and begin to address the issues as they come. So now moving on and putting the searchlight on adolescents, which is the area of my research and my interest. I just wanted to give you a flavor of what we are looking at in terms of the top 10 contributors of DALYs. And I'm sure most of you already know this, but just to reemphasize that alcohol is there, and it's prominently represented. It's on the 6th rank. But if you look at the other causes, road accidents, and many other causes that you can see on this list, it's clearly suggestive that there are some other alcohol links undertones to this. And so when you club this, you can agree with me that it probably would be the first, the most important cause of DALYs in adolescents. So then the question is, where is this happening? Where are adolescents drinking most? And for the purposes of this talk, I decided to look at the regional distribution of what, and based on the very latest global status report on alcohol, 2018, which was published a couple of months ago. And what we are seeing here, and what I'm flagging up here, is that the Southeast Asian region is it's quite it is becoming problematic in terms of how our young people are drinking. It's second to the Western Pacific region. And so that already tells us, and the review, the report clearly says that with the projections of 2025 of how things are going to pan out, it says very clearly that the Southeast Asian region is going to be the main driver of alcohol problems in the world. Whilst we are seeing a decline in other regions, we are seeing an increase in the Southeast Asian region. So clearly, this means that it's definitely of public health concern, and we should begin to do things to see how we can prevent that from escalating any further. The sad picture is that even with these stark realities of the burden that I've just tried to put out there, it's not being matched with how we are handling it and how we are providing care. And so there's this yawning treatment gap, and it's so massive that I'd wondered what you would guess. But there's an interesting slide that anytime I talk about it, people wonder. They always think that, as we have always said, it's beginning to be a very common password that most of the problems are in Islamic settings. But what we are seeing here is that when it comes to alcohol problems and many other even mental health problems, the treatment gap, which is that the number of people who have a case or who have a disorder but do not get the required treatment, that gap is almost comparable to what you find in low and middle income countries, so just a shade above that. In India, it's up 85% for alcohol problems. And that is not surprising because India has less than one mental health professional per every 100,000 population. And so that is not surprising. And, but with this data, we need to start thinking innovatively, and that's what I said in my introductory remarks. What do we then do? How do we go about this? WHO usually will show leadership in most of this, and so WHO came up with their strategy to deal with this. And WHO's strategy was to introduce what we call the Mental Health Action Gap, which gave birth to the Mental Health Action Intervention Guide, which is essentially a guide that primary health care physicians will be able to use. You just basic training, a few weeks training of it, and you'll be able to use to provide, to detect, to screen, and then provide immediate care to people who turn up with mostly common mental disorders. And that also includes alcohol use problems. Featuring very strongly in the mental health action gap is the role of psychological treatments. And psychological treatments, as the name suggests, are just treatments that, one of my colleagues said very simply, they're talking therapy. So it's treatments that use talking techniques and try to change people's emotions, thinking and feelings and behaviors, and try to help them deal with their interpersonal relationships with a goal to attain recovery. And there are these examples of psychological treatments that are now evidence-based. So CBTs and behavioral activation, interpersonal psychotherapy. These are all great, and that has come up because of years of very good accumulating evidence about their efficacy and actually effectiveness as well. So we do know that psychological treatments do work, and they are very attractive because most people probably would prefer that than to get a pill or a jab. So that is something that we in global mental health have been trying to further in terms of providing treatment. An offshoot of psychological treatments is what we call behavioral brief interventions. Brief interventions are specifically directed at in the alcohol field, in the alcohol domain. They are specifically used to treat alcohol use disorders. And they are targeted at non-treatment seeking populations. So essentially, you just turn up at the hospital for some other condition or the primary care they are delivered mostly at the primary care anyway. And you turn up for some other condition, and the doctor uses that opportunity to have a very quick, brief chat with you about ask pertinent questions about your lifestyle, including alcohol, and then gives you advice. And what we have seen is that that type of treatment or those brief interventions have really been very, very effective in helping people deal with their drinking problems. And so there's lots of interest now in that. I mean, if you can really do something in a very brief period of time, and it's working well, why don't we see how we can capitalize on that and use it? The problem is that it's not that big of a problem, but the only thing that I tend to differ, and that's what is informing my work going forward, is that there is a certain dominant treatment model, or the most frequently treatment model that we are used to, which is usually the face-to-face. So there's always the very typical biomedical approach to handling cases or dealing with, so patient-doctor relationship. So you need someone, and you have to talk to them on a face-to-face. And that is what has been with us from time immemorial, I guess. But of recent, there has been some very interesting, provoking discussions about that. Alan Kasdan, a very, very renowned Secretary of Research, is beginning to question and asking if we really want to make mental health treatment accessible, which clearly at this point we are not really scratching the surface that well, then we need to begin to think innovatively about doing something different from the face-to-face thing, because that may not work. And these are very interesting arguments that I tend to side with, which is, I just described what it is, the treatment sessions that are provided in person. You usually need to have a highly skilled or trained mental health professional. And now I can add that even with tax sharing and tax shifting, we have been able to redistribute very minimal tasks to people with less qualifications. But even with that, you still need counselors. You still need those lay counselors. Sessions are usually held in a given place. You have to move to a clinic, or someone has to move to you. So I think that these, even though they're great and I think they have helped us all these years, but I think that if we really want to think about how to then get mental health services out there, there are challenges, and the challenges that I think this comes up with, this obvious one comes up with, is that access to treatment is a problem if you continue with this model, because obviously we don't have enough. That the simple truth is that we don't have enough. I just said India has less than 1 in 100,000 mental health professionals. We don't have enough to handle the population. Then there's also the issue of the availability of distribution of professionals. So we do know that mental health professionals, some people specialize in child psychiatry and many other things, which are very, very well sought after. But unfortunately, we don't have many of those specializations in the field. So then it means that where there is a huge need, we really don't have the matching professionals or services for them. So that is what, and the last obvious challenge with this model is that obviously, the whole stigma thing. I know how mental health is supposed to be a whispering discussion in most places, and so then to have people going out there to actually go seek help and be seen as probably having a mental health condition may be counterproductive. So, because of that, we in Sangad and for this particular fellowship, I thought very strongly. I mean, obviously, the public health burden is made, the case for interventions is made, but then how do we then deal with the issue of the treatment gap, and how do we, especially for an adolescent population, how do we go around this issue of given what I've just laid out there in terms of the challenges with the dominant model. So I then said that, well, one thing we could do is let's leverage on technology. Let's sit down and see if we can reach them remotely. Obviously, there are, so my key goal is to address this treatment gap, delivered through technology. And what I am beginning to these days is becoming a buzzword in most of my talks is what I'm beginning to call digitization of tax shifting. So, as I said earlier, tax shifting is traditionally still delivered. Even though tax shifting, you are definitely redistributing the smaller tasks to people who are less qualified, so you're able to get more people to do it. you still do need a human factor. You still need a human factor. I'm not in any way suggesting that the human factor is not important in psychotherapy, but I'm saying that if we do want to really reach many people, we need to start doing things innovatively. And it may not just be one model. It may be combining models. So one of the things that I'm thinking about is to begin to think about how to digitize tax shifting. And that is what my fellowship is about. And the case, particularly for why I'm doing this, I've already laid it out a bit, but just to reemphasize the case in India, that in India, the minimum age of initiation has dropped. This is very, very cautionary note. This is a study that was done in just one state. But what we did find there was that it has dropped from 19 to 13 years. So the young people are beginning to go out there and start drinking much earlier than it was the case in 20, 30 years ago. And about 20% of them drink in risky patterns that suggest hazardous drinking. So my work is focusing on hazardous drinking. And I know a colleague, I was just chatting with a colleague over coffee the last time, and I needed to clarify that we are dealing with the less severe. It's also a concern because if you, like I said in my last bullet point, if you don't tackle it now, it's likely going to have consequential effects in future. But that is where you find more people actually with that problem. So we are tackling hazardous drinking. It's not harmful or dependent drinking. It's not those people who have drinking problems, as in they are dependent on drinking. We are not tackling that in adolescents. We are tackling hazardous drinking, which has potential causes for drinking problems later in life. And it's interesting when we started doing this work, before I put in this whole thing, it was all about reading up the literature and knowing what is going on in India. But when we started, we got more validated in terms of the work we wanted to do when we started seeing things like this popping up in Goa, where we have adverts for drinking competitions. And your guess is as good as mine who the target group would be for this. That's very worrying. And so, as I said, BI's brief interventions are great. They are efficacious. They are able to reduce drinking by about 23 grams in adolescents in some studies done in the West. But we don't know how they are generalizable in our setting, 1, 2, among adolescents, and even delivered over technology. So 3 questions that we think are very relevant to want to answer here. And so that's why we're saying that we don't know, we don't have comparable data in our setting. So our question is, can PowerText, and what we call our project is, our intervention is called PowerText. That's our running popular type name of the fellowship is PowerText. And essentially it's trying to harness the power in a textual form to see whether if you use that to deliver a brief intervention for adolescents aged 18 to 24, whether that would be able to reduce hazardous drinking. And more importantly, what we want to find out is the mechanisms of change. Most of these studies that have been done have just been able to, let's say, more cross-sectional descriptives of what they found, but we want to go beyond that and be able to find out what really created this change, what made and what were the elements of the psychological treatment that helped make the change. So we came out with a design. So our design is an intervention that is phasic, and it's also delivered, looked at in terms of sessions. So we have 3 phases: the orientation phase, personalized feedback. The adolescent will get personalized feedback and try to orient the adolescent at the start. And then the next phase of it is to set some goals, and then to try to learn some skills. And then the last phase is to be able to build skills to maintain that. And then we are going to have an interactive element to it where obviously it's a chatbot, so they are going to be typing and getting some feedback from the robot, and it's all personalized action plans that are also included. So the features, we have 4 main components or features of our intervention, PowerText. So we are still coming up with a name for our chatbot, but we are looking at a chatbot being able to deliver messages. We're going to use animations. We're going to also use infographics and videos to try to change behavior. There's no time now. I've just been told that I have just 5 minutes, and so I will skip. I wanted to show you a video clip, just a trailer of one of the clips that we have prepared. but there's no time. One thing, the key lesson so far is that we think that there are definitely quite interesting similarities between drinking problems here, how adolescents drinking behavior here and in other places. The only thing that probably stands out is that there seems to be some stronger cultural norms here, which we think we are going to capitalize on for our intervention. It's always good to think forward because we are beginning to think about how this can potentially be scaled up if we do find an effect. And we did rely so much on our data. So one of the key, hopefully what we hope to get out of this is by the time we finish this, we should be able to have a methodology for delivering brief interventions over mobile messaging. And hopefully there is a program that the Indian government has just introduced, the RKSK program, which has emphasized this. So we hope that we will be able to contribute some useful data in that program. How many minutes do we have? 2 minutes, okay. Okay, we just want to talk a bit more about one of our things, there's no time, Lydia, sorry about that. I wanted Lydia to talk about the public engagement thing, so I will just skip about that. But we just got a public engagement grant to support our work for the fellowship, and which is very useful. So I'll just skip that. But one of the things I was also asked to talk about in this is that to share with you how this fellowship has helped me. Three key things. The mentoring has been amazing. The whole mentoring arrangement for as an intermediate fellow, you are supposed to have a mentor, and that has really, really helped me so much. And it's gotten me to be able to have to even translate those benefits into Sangat, where I've set up a mentoring scheme, a mentoring system in Sangat, where we have picked up international people to mentor people in Sangat, potential high flyers in Sangat. So that got me to be able to, motivated me to be able to do that. Amazing networking opportunities with this fellowship. I mean, the last couple of months ago, I think in May, we were in Joburg with under the invitation of Deltas. And through that, I'm beginning to talk to colleagues in Africa to see how we can collaborate on research projects. Obviously, I'm beginning to be asked to sit on and get more, collaborate on research grants that are difficult to accept because of my role, my time on the fellowship. But that is clearly what is beginning to see, and I'm currently giving some mentoring support to some of the students also. So this is just my team. I introduced some of them earlier on. This is my core team and my field team, and then Lydia here, my intern, who has come to join us for a couple of months. And I just want to end by saying, Dhanyawad. Did I do well with my...
Thank you very much, Ben.
Yes.
So in the interest of time, I think we have to take only 2 questions, brief questions. One over here.
Vaibhav, wonderful presentation. I work on chronic diseases in rural Gadchiroli, and alcohol is a big risk factor. You said that one of the comments you made was BIs don't seem to work in low middle income countries. Can you please elucidate on that a bit?
No, so I'm saying the evidence-based for their effectiveness is not that well established in low and middle income countries. I think you can say the jury's out for high income countries, but not in low and middle income countries.
What's the typical endpoint that is, what's the primary outcomes have been used for BIs? Is it change to non-harmful drinking?
Yes, so it is about reducing. So it's more about reduction, and so it's not to get them to absolutely stop it. It's more about reduction.
And what's the typical effect size?
Interesting. So that's what I just said. That in one of the meta-analysis that we cited, that had mostly high-income country studies, they were able to reduce it by about 0.25 of the standard deviation. So it's quite small. But I guess the question is that if you can achieve this with a larger, I think it's more about the coverage, and that's what we are trying to capitalize on. So even though the effect sizes may be small, if you're able to get it to more people, at the end of the day, it makes public health sense.
One question there. One last question.
Thank you. Great presentation. I just wanted to know, do parents confound your BI, or do they actually help in this process?
Parents?
Parents.
Right. So when it comes to adolescents, and especially adolescent drinking behavior, the theory and the framework suggests very strongly that parental involvement could either be a positive or a negative. It's one thing that you have to address the environment of the adolescent, where the adolescent lives. So for instance, what the adolescent sees around. If he sees a certain behavior in a parent, he may, modeling is very, very, very common. But the adolescents are also very, very private people. They want to, when it comes to that, they want to keep things like that to themselves. So in fact, that was one of the reasons why, someone was going to ask me why we started at 18, but one of the reasons why we did that, I mean, apart from the fact that the burden increases as they age, the older adolescents, is that we were really concerned about the potential for this to create trouble at home when the parents get to know that their adolescents are drinking. So we struggled with that a bit, and then we thought, well, we are not, at this point, we don't really have the answers to that. We will definitely think about answers for how to tackle this later. But yes, parental involvement can be either good or bad, yes.
Okay, thank you very much,
Okay.
Ben,
Thank
for
you.
a very interesting talk.
Thank you very much.
Okay, our next speaker is Dr. Arun Shukla, who's an associate professor at IIT Kanpur. He's recipient of EMBO Young Investigator Award and the Intermediate Fellowship from IndiaAlliance. Arun's interest lies in studying structural and function of GPCRs. Over to you, Arun.
Thank you, Sujata. It's an absolute pleasure to be here. As we are celebrating a decade of IndiaAlliance, I thought I'll give you a brief overview of the research program in my lab that we have established in the last nearly half a decade. And I'll not bog you down with details of experimental data, but I'll be available in lunch if anybody wants to talk about specifics. So we are primarily interested in a family of receptors called G-protein coupled receptors, GPCRs, and they constitute the largest family of receptors in our body, about 1,000 different members. They're also referred to as 7 transmembrane receptors because they span the membrane 7 times. And about 200 or 250 of them are functionally known, while the remaining receptors still remain orphan. And these receptors are involved in every physiological process that you can think of, starting from brain function, behavior, cardiovascular regulation, pain sensation, immune response, and so on. And thus, it's not surprising that about 40% of the currently prescribed drugs that target GPCRs. For example, angiotensin receptor blockers are prescribed for hypertension, histamine receptor antagonists for allergies, long-acting beta agonists for asthma, and so on. Therefore, it's very important to understand how these receptors work. That is, how they recognize ligands, how they're activated, how they signal, and how they're regulated, not only to understand the basic aspects of cell signaling, but also in long term to utilize that information to design better therapeutics. So I'll take you through the current paradigm of how these receptors work based on several decades of research. So binding of an agonist, which is an activating ligand, leads to a conformational change in the receptor and its activation, followed by coupling of heterotrimeric G proteins, and hence the name G-protein coupled receptors. This, in turn, results in generation of second messengers, such as cyclic AMP, inositol phosphate, calcium, leading to what we typically refer to as G-protein dependent signaling. But obviously, cells have to control the signaling in a very precise manner, and for that, they have designed a mechanism called desensitization. Essentially, what happens is that activated receptors are phosphorylated primarily in their carboxyl terminus by a family of kinases known as GPCR kinases or GRKs, and that leads to interaction of a multifunctional protein called beta-arrestin. You'll be hearing more about this in today's lecture. And it is believed that interaction of beta-arrestins essentially comes in the way of G-protein coupling. That is, they sterically hinder further G-protein coupling, leading to termination of G-protein signaling response. Now, this has been the classical paradigm. However, in the last 10, 15 years, it has been discovered that beta-arrestins can do many additional things. For example, they can interact with a number of components in endocytotic machinery, such as cladribine and adaptin, to mediate receptor endocytosis, which is basically the process of receptor trafficking from the cell surface to inside the cell. They can also scaffold E3 ubiquitin ligases to bring them in close proximity of the receptor, thereby mediating ubiquitination and degradation of the receptors. But most importantly, they can also scaffold a number of kinases and phosphatases to yield what we refer to as G-protein independent, beta-arrestin dependent signaling. So essentially what it means that, unlike what is there in the textbook, the signaling downstream of GPCRs is bifurcated. It can happen through G proteins or it can happen through beta-arrestins. And an interesting aspect of this is the cellular and physiological outcomes downstream of G protein signaling or beta-arrestin signaling. They're very distinct from each other. And that has huge applications because in the past few years, it has been possible to pharmacologically separate these two signaling pathways. And I'll tell you what I mean by that. So when an antagonist, which is a ligand which inactivates the receptor or puts it in inactive conformation, none of these signaling pathways are triggered. When a full agonist or unbiased ligand or endogenous ligand for a receptor, when it binds to a GPCR, you get both arms of signaling G proteins and beta-arrestins. But it is possible to design ligands which will bind to the receptor and selectively trigger either beta-arrestin pathway or G-protein pathway. And these ligands are referred to as beta-arrestin biased or G-protein biased ligands. And it turns out that many drugs that are prescribed that work on GPCRs, their side effects and their desired effects are going through either the G-protein or the beta-arrestin pathways, meaning the desired effect and side effects are separable from each other. And this has sort of led to the concept of biased agonism that has implications for novel therapeutics, and I'll just give you one quick example. So morphine, which is taken to manage post-surgical pain, is basically an unbiased ligand for mu-opioid receptor, or GPCR. It's very effective analgesic, but it also leads to undesired effects of constipation, respiratory problems. It turns out that the pain relief is going through G-protein pathway, while the undesired effects are going through beta-arrestin pathway. So now if you think about it, if you can modify morphine or design new analogs or identify new ligands from scratch, which are G-protein biased ligands, if they bind to the receptor, they will give you pain relief that you want, but not the side effect. And hence, what you have is a more effective therapeutic, so to say. And this is one example. There are multiple examples across many different receptor systems. In some cases, it's the G-protein signaling that gives you desired effect. In some cases, it's beta-arrestin signaling that gives you the beneficial effects. And now this is changing the landscape of drug discovery in major pharmaceutical companies that are focused on GPCRs, and many of the biased ligands are in different phases of clinical trials, including one that is in the final stages of approval by FDA. So receptor G-protein interaction has been relatively well understood, but receptor beta-arrestin interaction and the subsequent regulation of the receptors is lagging behind, and that is where our focus has been in the last 4.5, 5 years or so. So this interaction of receptors with beta-arrestins is a biphasic process. So what happens in the first step, beta-arrestin, which is shown here in blue, interacts with the carboxyl tail or carboxyl terminus of the receptor, shown here in yellow, and forms what is referred to as partially engaged complex, or also referred to as tail-engaged complex. And then in the second step, beta-arrestins engage further with the transmembrane bundle of the receptor, leading to the formation of fully engaged complexes. And in the field, it has been generally sort of believed that the fully engaged complex is the one that mediates all the functions of beta-arrestins, and the partially engaged complex is simply a passive intermediate. But when we started, we didn't sort of buy this notion, and we used a combination of pharmacological, biochemical, structural approaches to assemble these two types of complexes in vitro and in cellular, and ask the question whether the partially engaged complex might also have some functional relevance. And turns out that, in fact, there is a functional separation in these two conformations of receptor beta-arrestin complexes, where the partially engaged complex is competent to mediate endocytosis and signaling, while the fully engaged complex is driving the desensitization. And when you think about it, it makes sense, because for desensitization to happen, beta-arrestin has to snuggle against the receptor so that it can occlude the surface on the receptor, which was otherwise being occupied by G proteins. And partially engaged complexes can drive endocytosis and signaling, because beta-arrestin, even in this conformation, can interact with clathrin, can interact with ERK-MAP kinases, and so on. Now, since we published it, 3 other groups across the world, they have, using different receptor systems, different approaches, they have converged to the same notion, suggesting that this is a new paradigm in these complexes, how these complexes work. Interestingly, what we have also found is that if we take a beta-arrestin bias ligand, which upon interaction with the receptor mediates only beta-arrestin signaling but not G protein signaling, it exclusively forms only the partially engaged complex, and there is no formation of fully engaged complex. And it makes sense, because in this case, when a bias ligand binds to the receptor, there's no requirement for desensitization, because there's no G protein coupling in the first place. Whatever is required of the ligand, that is endocytosis and signaling aspect, can be mediated by partially engaged complex. So it provides a previously lacking mechanistic insight into biased signaling and how biased ligands mediate their effects. Now, what I have told you so far, basically this paradigm has been developed using synthetic ligands or receptor mutants. So a question is whether, if we can sort of design and understand it in vitro, is it possible that nature has already designed it in our system? We have just not identified them, meaning, is it possible that bias ligands, bias receptor systems might be available or might be present in our bodies? And when we look to the literature, it turns out that there are many 7-transmembrane receptors which are classified or which are still present in the GPCR superfamily, but because they have 7-transmembrane architecture, a typical signature of GPCRs, but they do not couple to heterotrimeric G proteins, and hence they're basically classified and somewhat ignored as non-signaling or non-functional receptors. But if you knock them out, you get very specific phenotype, suggesting they must have some functional role. Intuitively also, it doesn't make sense that nature would design something that's sitting in the membrane, taking space and doing nothing. So we basically, and at the time when they were classified as non-signaling receptors, the beta-arrestin signaling was not appreciated that much, and hence people thought the only way these receptors can signal is through G proteins, and hence they were classified as non-signaling receptors. So we have basically connected these 2 dots and asked the question whether these so-called non-signaling or non-functional receptors, might they be signaling through alternative pathways, perhaps beta-arrestin pathway? And in fact, what we have found is, as present in the literature, these receptors do not couple to heterotrimeric G proteins. They are basically flat in G protein coupling assays. But they do very robustly recruit beta-arrestins. Not only they physically interact with beta-arrestins, but they're also getting internalized in beta-arrestin dependent fashion, meaning they are in fact beta-arrestin coupled receptors. What about their signaling? And to identify, to discover whether these receptors might signal, and if they do, what signaling pathways might be involved, we have used antibody array-based approaches or global phosphoproteomics analysis to identify different proteins which undergo phosphorylation, dephosphorylation, and these receptors are activated. We have identified a number of proteins. Some of them fall in the previously known category of beta-arrestin signaling pathways, and we have identified some unique pathways as unique proteins as well. And this phosphoproteomics was done with vProteomics, a venture in Delhi that is started by a former IndiaAlliance fellow, Gagandeep. And in the past, we have worked with top-notch ventures in the world, and the data quality, the services that they provide is on par with international ventures. So we are very happy sort of in working with them. And once we have identified these signaling proteins downstream of these beta-arrestin coupled receptors, we have utilized basic bioinformatic analysis and then identified new signaling pathways that some of these receptors can signal through. And we have experimentally validated some of them using inhibitors or knockdown approaches. Interestingly, when we reconstitute beta-arrestin complexes of these receptors using stabilizing antibody fragments, what we find is that these receptors exclusively form this partially engaged complexes with beta-arrestins. So it's coming back to the biased ligand scenario, where biased ligands induce only partially engaged complex. Similarly, these receptors bound to their native or endogenous ligand, they also induce formation of only partially engaged complexes. And this makes sense, again, because similar to biased ligands, there's no G-protein coupling for these receptors, and hence there is no need for desensitization, and therefore there is no requirement for the fully engaged complex. Now, going forward, we are using cryo-EM and X-ray crystallography to get high-resolution structure of these receptors to see, despite having 7 transmembrane architecture, how they can display such a unique functional divergence compared to prototypical GPCRs. Now, I've been using the term beta-arrestins, plural, because there are 2 isoforms of beta-arrestins, beta-arrestin 1 and 2. And if we superimpose their structures, their 3-dimensional structures, if these were not labeled, even I cannot tell which one is beta-arrestin 1 and which is beta-arrestin 2, because they're structurally very similar. They have about 78% sequence identity. Interestingly, it turns out that they have, despite having structural and primary sequence similarities, they have a very significant degree of functional divergence. And what I mean by that is, for a given receptor, it's possible that beta-arrestin 2 is predominantly mediating endocytosis, while beta-arrestin 1 is mediating signaling, or vice versa. So that is one receptor, 2 isoforms of beta-arrestins doing different things. There are also cases where the same isoform of beta-arrestins can mediate very distinct signaling pathways for different receptors. So how does it work? And this has been a huge mystery in the field. And we have basically sort of utilized, once again, a combinatorial approach of structural, biophysical, biochemical sort of techniques, including spectroscopy, negative staining EM, to identify that the 2 beta-arrestin isoforms, that is beta-arrestin 1 and 2, when they bind to the same receptor, they can adopt very distinct conformations. And in fact, these distinct conformations, in turn, drive their functional divergence. So we can make chimeric beta-arrestin constructs, and we can encode new functions in them. What I mean by that is we can make beta-arrestin 1 look more similar functionally to beta-arrestin 2, and vice versa. So there's distinct conformations of beta-arrestins that they can adopt when in complex with a GPCR is driving their functional divergence. So this has come back with these interviews. We are hoping to send it back in a few weeks. What about same isoform doing different things for different receptors? And to probe that, we have designed antibody fragment-based sensors that can selectively recognize receptor beta-arrestin complexes in vitro and in cellular context. And I'm just giving one example here. So in green, you basically have an antibody fragment that is expressed as an intrabody. An intrabody is basically when you express an antibody as cytoplasmic protein. And the intrabody is tagged with YFP. Beta-arrestin 1 is tagged with red and cherry. And this intrabody has specific recognition for receptor-bound beta-arrestin 1. So what we see here is that under basal condition, they're both cytoplasmically distributed, as expected. When we hit the cells with agonist, beta-arrestin from cytoplasm goes to the membrane, where the receptor is sitting, so that it can interact with the receptor. And intrabody very faithfully follows the receptor to the membrane. Upon longer agonist exposure, the receptor beta-arrestin complexes get internalized in these endosomal vesicles. And once again, the intrabody sensor follows the complexes in these endosomal vesicles, suggesting it's a good sensor of receptor beta-arrestin interaction, their trafficking, and the complexes that are formed in the cells. Now we have used this intrabody sensor and looked at about a dozen different GPCRs. And turns out about half a dozen receptors behave the same way. That is, they form a type of complex or a conformation with beta-arrestin that can easily be recognized and followed by the intrabody sensor. But then the other half a dozen or so receptors, including, for example, bradykinin receptors, they show a very distinct pattern. So in this case, bradykinin receptor is a prototypical GPCR, like vasopressin receptor, which was used here. Again, upon against stimulation, beta-arrestins go to the cell surface, and then they get internalized in these endosomal vesicles with the receptor. But unlike vasopressin receptor here, the intrabody is sitting pretty in the cytoplasm even after agonist stimulation, suggesting that the complex of beta-arrestin 1 with bradykinin receptor must be conformationally very distinct from the complex of vasopressin receptor beta-arrestin 1 complex. So this sort of provides a mechanistic insight into how same isoform of beta-arrestins can mediate different functions for different receptors because they can form different conformations with these receptors. Now, my talk title said structure, function, and modulation, so I'll talk about modulation in 2 minutes. So, as I said, the driving force for the functional multiplicity of beta-arrestins has been their ability to interact with a number of cellular partners such as clathrin, ORC-MAP kinase, JNK, CSORC, and so on. So we have, and if you look at the interface of these interactions, they are very distinct on beta-arrestin's surface. And therefore, we wondered whether it would be possible to selectively interfere some of these interactions and thereby selectively take out one function of beta-arrestin while not affecting the others. If you think about siRNA knockdown, knockout approaches, CRISPR-Cas, they basically lower the level of protein, such as beta-arrestin, lose all the functions across the board. We wanted to selectively take out one function while not affecting the others. So again, we have used phage display-based technology platform to generate antibody fragments against beta-arrestin 1, beta-arrestin 2, and so on. And I'll just give you one example. We have identified a particular antibody fragment that very selectively inhibits the interaction of beta-arrestin with clathrin. It does not significantly affect the interaction of beta-arrestin with ORC-MAP kinase. When we express it as an intrabody in cells, it can very efficiently inhibit the endocytosis of vasopressin receptor, which is a prototypical GPCR, shown here by whole cell ELISA, also by confocal microscopy, where in case of control antibody fragment, the receptor is internalized in these endosomal vesicles. In case of cells expressing this inhibitor of beta-arrestin clathrin interaction, receptor beta-arrestin complexes remain tethered to the membrane even after prolonged exposure. So this has allowed us to separate the endocytosis and signaling from each other, which sort of changes the previous notion in the field that you must have endocytosis to get ORC-MAP kinase activation in beta-arrestin dependent fashion. So the earlier the idea was that you must have these signalosomes on the endosomal vesicles to get ORC-MAP kinase activation. But using this antibody fragment as a tool, we have sort of defined that separately. And interestingly, as I said earlier, it has also been possible to use this antibody fragment to selectively take out endocytosis function without affecting ORC-MAP kinase activation. We have now antibody fragments which can do the opposite, that they can inhibit ORC-MAP kinase signaling without affecting the endocytosis. And more interestingly, we have identified antibody fragments that can allosterically stabilize these complexes in the cells, and thereby they can enhance, for example, the kinetics or the onset of signaling or endocytosis, and we can play with compartmentalization of signaling and so on. Hopefully, I'll be able to tell you more about that next time. So due to time constraint, I'll not talk about the summary, but I'll just, in 2 minutes, I'll tell you about the impact of IndiaAlliance as I see it. Since I moved to India, on a couple of occasions, I've been asked by different outlets to share my experience and why I decided to move back to India. And I've said what I honestly feel. If it was not for IndiaAlliance Fellowship Program, perhaps I would have not returned back to India in the first place. And even if somehow I did, perhaps we could not have done whatever little we have done in the last 1.5 years. But in my honest opinion, my personal opinion, I think the impact of IndiaAlliance is not about the success of individual fellows or a few fellows or some of us publishing glossy journals or awards and so on. But rather, the impact is that it has inculcated a belief, a faith, a confidence in us that despite all odds, we can do cutting-edge, globally competitive science in India. And I think that is the true impact. The other thing which I feel the Alliance has done is it has broadened the footprint of quality science across the country. So it's not the case that you get quality science only in localized pockets, but now it's spreading all over the country. What really gives me immense pleasure when I go to a Gordon meeting or Keystone meeting, when a very productive postdoc comes to me and says, I was thinking about setting up a shop in States or Europe, but now after seeing what India Alliance fellows can do in India, I have decided to apply in India and look for a faculty position there, and that is what makes me ecstatic. Obviously, the last decade has been phenomenal of India Alliance, but I can say with full conviction that the next 10 years are going to be even more productive, more exciting, and take the Indian science to a whole new level. As they say in Hindi, kahani abhi baki hai. Thank you.
Thank you, Arun, for a very wonderful presentation and very kind words. So the hall is now open for questions. We can take only a couple of questions in the interest of time. Professor Shankar.
Thank you for this really interesting talk. So, have you looked at the classification of this non-canonical GPCR or beta-arrestin signaling across different cell types to see if they are A, cell type specific, or if they are a given function, phenotypic function? I'm not talking about phenotypic function in terms of ERK signaling. I'm talking phenotypic function in terms of the endpoint consequence in cells. Have you looked at the classification of these under relevance and cellular function and normal and pathological function?
Sure, absolutely. So that's a wonderful thing, and we are sort of going in that direction a little bit. For example, the signaling pathway and the cellular outcome that we have identified for one of the chemokine receptor D6 is, it seems to be in a PKD, AKD, coffulin pathway in migration. And these receptors are expressed in those immune sort of cells where migration is one of the key functional outcomes. So that does seem to make sense. But we have not sort of looked at the entire family of receptors yet, because the idea, at least in the beginning, was to establish that these are indeed beta-arrestin signaling receptors and then see how wider, how many other receptors will follow the same pattern. So going forward, we'll definitely be looking at that.
Okay, one last question there.
I have a short question. See, how stable are these intermediates and physiologically? And your antibody fragments are actually interfering with its stability when you track the protein in or out?
Right. So that's a very good question. In fact, for structural studies, we have to stabilize these complexes, even the fully engaged ones, with antibody fragments. Otherwise, we cannot really capture them by crystallography or by cryo-EM. But that is the beauty of the biased ligand and the biased receptors, where all that we are getting in cellular context as well, without any antibody fragment stabilization, is only the partially engaged complexes, meaning that these are reasonably stable. If you compare them side by side with fully engaged in terms of kinetics and stability, that we have not done, but they seem to be reasonably stable where they can mediate the functions downstream.
Okay, thank you, Arun. And I'm sure you all can discuss offline with Arun. So the next speaker here is Uma Ramakrishnan. Professor Ramakrishnan is working at NCBS. She's a recipient of India Alliance Senior Fellowship. She's an associate professor at NCBS. Her research work focuses on understanding the impact of genetic variation on the well-being of tiger population. Over to you, Uma.
Thanks all for staying, and I'll start off by thanking the India Alliance. You must be wondering what someone who works on tigers is doing at a biomedical venue, but I guess the questions I'm choosing to address are about inbreeding. In this case, I'm trying to look at the consequence or potential consequences of inbreeding and genetic variation on fates of tigers instead of maybe humans or flies. So that's another 2 minutes off of my already 30 cut to 20. No. Great. Thank you. Yeah. So as I said, when I started off, that I'm interested in inbreeding. Oh, sorry, I did something wrong. Oops. which one is the pointer? This one. So inbreeding is basically mating between relatives or consanguinity, right? And we've known for a long time, theoretically, empirically, experimentally, that when we have inbreeding and high inbreeding coefficients, which means more consanguinity, then fitness or survival actually declines. And this decline in fitness when an individual is inbred versus outbred is called inbreeding depression. And it's thought to be caused by what's called inbreeding load or accumulation of mutations and expression of recessive mutations in phenotypes. Traditionally, inbreeding has been estimated using a pedigree approach. That's what makes sense, right? You look at how related the parents of an individual were. But more recently, we've been able to try and understand whether there are other ways to estimate inbreeding. So for example, we now know from a lot of work on humans that whole genome data and these long stretches of homozygosity are a potential alternative way to estimate inbreeding. So what does this actually mean? So basically, for example, if you have parents who are closely related, their offspring tend to inherit long stretches, and this is basically very recent inbreeding, right? So it's not ancestral inbreeding or inbred founders, but if parents of an offspring are related, then the individual inherits these stretches of DNA which tend to be homozygous, okay? So there's a proxy or a different way to actually look for inbreeding without actually knowing about mating patterns and so on for individuals. There's also data for humans, for example, which shows quite well that there are several groups which have long runs of homozygosity. There's been some recent papers from India as well. And these long homozygous runs tend to harbor deleterious alleles which can have potential impacts on fitness. There's been work on other species as well, showing that these long stretches of homozygosity correlate with known inbreeding coefficients. So here you see basically this genome-wide estimate of inbreeding from these runs of homozygosity, but they correlate well, those estimates correlate well with pedigree estimates, and they also show strong impacts on survival and reproduction and so on, various traits. So I'd like you to keep in mind here that actually when you have inbreeding coefficients of 0.15 or higher, there seem to be significant impacts on fitness. Okay, sorry, I'm going back and forth. So where do tigers come in? So basically, like many endangered species, they have declined in numbers and also lost their habitat. While India boasts 60 to 70% of the global population, individual populations are actually small and several are isolated. So I guess the question that naturally emerges are what are the genetic effects in these small isolated tiger populations. So what I'm going to do today, I don't think I'll have time to go through all of this, is to talk about the inbreeding scenario of global tiger populations, give you a quick snapshot of that, and why I've decided to focus my particular study on this population, Ranthambore Tiger Reserve, very close to here in Rajasthan. Try to convince you that we've partially answered this question about whether tigers in Ranthambore are indeed inbred, and talk to you a little bit about ongoing work. I probably won't have time to get to this, another story of an isolated population, but let's see. So this is just very quickly a distribution map. It's not very clear of tigers around the world. Yeah, so these kind of light green patches, and this is a study which is close to being finished, but we've sequenced several genomes across tiger range, and they're sequenced at different coverage. And basically, when we look at variation across these different wild tiger populations, the ones in brackets are if they're from the zoo. What we find is really interesting. Of course, as we expect, there are different groups of tigers, genetically defined groups, not defined a priori by geography, but simply by genetics. And the most likely outcome is that there are 4 groups of tigers, and of course, Indian tigers are a separate group, quote unquote. And then when we further look at genetic variation within these groups, we find, as you might expect, this is distinct alleles and this is distinctive alleles, private alleles and distinct alleles, that, you know, if you do a rarefaction analysis, Indian tigers have the highest amount of genetic variation compared to other subpopulations. Okay, so that would then kind of belly what I just said earlier, that we should be concerned about genetics of Indian tigers, right? On the other hand, though, when we plot these runs of homozygosity, and this is basically on the y-axis, the proportion of the genome which is in a homozygous stretch. So you add them all up, okay? And on the x-axis is just different individuals colored by the subpopulation or the population. This particular individual is an inbred zoo tiger, so it has a high, so this is equivalent to your inbreeding coefficient in a sense. And I just drew a line at 0.15 because that's where we see fitness effects. You do see that while Indian tigers had high total variation, individual Indian tigers do seem to be inbred. Okay. So is this true for all tiger populations, or is it more true for some versus others within India? So just, I know many of you have probably seen this, but why are tiger populations in India isolated? So this is India in 1800, 1900, 2000, right? Sorry, 1900 and 2000. So clearly, there's been a lot of habitat loss, and this has resulted in fragmentation. And here you can see in black the tiger reserves which kind of remain. So some areas, like say the central Indian landscape, has several tiger reserves which are relatively close by, and the Western Ghats, for example, but other areas, they seem to be relatively isolated. And today I'm going to talk about this tiger reserve, Ranthambore over here, which is basically looks to be physically cut off from other kind of tiger reserves close by. There's no connection here. This is actually an extinct population, Sariska, which was repopulated with Ranthambore individuals, and this one is the same fate as well. If I get time, I'll talk about Simlipal towards the end. Okay, so this is very bleached out. I'm sorry for that. But this is a study which we did earlier on to basically, again, within India, define what the populations are. And this is based not on whole genomes, but 10,000 SNPs. And this shows us, for example, two key inferences. One, that Ranthambore, which I said may be isolated, is definitely a distinct population genetically. And also, interestingly, if you just calculate relatedness between individuals in Ranthambore, they are on the order of siblings. They're very highly related compared to any of the other populations or protected areas we look at. Okay? So Ranthambore is potentially isolated and possibly inbred. So here's the data we've recently generated, and I'll again walk you through this. This is again the same statistic on the y-axis, proportion of genome with longer than 1 MB ROH, the total proportion, so added up. And here you can see this again kind of cutoff of 0.15 in breeding, and these are data from genomes, about 30 or so genomes from Central Indian tigers, Northeast, North, South India, and there you can see the Rajasthan or Ranthambore tigers, many of which seem to have a much higher level of ROH than other individuals in other populations. What about if we look at this from a time perspective? So clearly, they may have been founders, and then there's continued inbreeding over time. So do we actually see an increase in inbreeding as we are measuring it? This is a proxy for inbreeding, right? We'll get to real inbreeding maybe a little later. So what happens? So this is just a plot, and this is again preliminary results, which show this, maybe this axis is a bit confusing. This is birth year where this is present, okay? So this is an old individual, and this is a young individual. And there seems to be some kind of pattern, but I can't be sure. I think we really need more older individuals to comment more on this, but a very young cub does seem to have relatively higher runs of homozygosity than does an older individual. But maybe it's not that the whole population is getting more and more inbred. Certain individuals are in family groups where there's more inbreeding than others. So because Ranthambore is a very well touristed and visited population with active management, there's some kind of a putative pedigree. People say, oh, this is the mate of this one, and so on and so forth. And so can we actually incorporate that information and test this pedigree? So then we would really know who is inbred and who is not inbred, and then test it back against those runs of homozygosity if possible. So to do this, though, we need to follow specific individuals and collect their DNA samples. All the work I showed you so far is whole genome data generated from blood samples, which we get opportunistically in case an animal dies, in case an animal is moved or something like that, right? So how do I go and look for animals and sample their genetic material? That's actually what's most interesting anyway. So this is just to show you, for example, some parts of that pedigree. That was very messy, but you can see these are loops. Like this individual basically has mated with his daughter, and so there's definitely signs of inbreeding within this pedigree, should it be correct. And just to show you, we've already sampled these circled individuals, but how have we sampled them? So I've actually been doing this for a long time now, working on wildlife populations, and I've mostly worked with non-invasive samples, which are fecal samples. So my go-to initially was, oh, maybe we should just sample fecal samples from these individuals. This is another example of a pedigree. So I guess one of our questions then was, how do we sample genomes from identified individuals, and which sample source provides better data? So first of all, if you follow an individual, what kind of sample do you get the most of? So this was really surprising to me. We followed individuals in Ranthambore for about 1.5 years of field work on field, and basically what we find is that most often we are able to acquire shed hair samples versus something like scat, or of course if an animal dies and so on, right? So basically when these animals sit down, they rub against the ground or bushes and so on, and once they leave, we search the site and we can find hair. But is hair actually a better source of DNA than fecal samples? Maybe we get fewer fecal samples, but they're better. Actually, it turns out that fecal samples are really poor. They're a good source of DNA, but very poor source of DNA if you want to get only tiger DNA, right? There's a lot of other stuff in them, prey DNA and so on and so forth. And so if you just whole genome sequence DNA extracted from shed hair, of course tissue is 100% tiger. This on this axis, sorry, I lost the axis, is proportion of reeds that map to tiger. But feces on the other hand is almost zero. So there's a lot of variation for hair. Some samples give very good results of very high mapping, whereas others are relatively poor. But still right now, hair is our best bet. And we've already sampled 30 individuals in a population about 60. So that's pretty amazing to be able to cover a carnivore population so well. So basically, that's what we've been able to accomplish so far. We watch these guys. They sit down. After they leave, we search the site for hair. And these are really small. So that's just to show you in the context of a 1 rupee coin. We collect these hair and then rush back to the lab and figure out what we can do with them. So this is just to show you, like the photo that was over there, that they scratch and we get the hair and we can actually reconstruct maternity. We already have had some surprises revealed, kind of defying the putative pedigree. So this is all fine, but our results are only as good as the reference that we compare our samples to. And so far, there isn't really a good tiger reference genome. The genome which exists is based on short-read Illumina sequencing, and that was very unsatisfying. So it also turned out that while we were doing this work, just before we started actually, this iconic individual, her name is Machli, she lived for 20 years and she had many, many offspring, so she's also a founder in this population. A very important individual died, and we were able to, we had permits, we were able to collect tissue samples from her. So we thought, why not use this opportunity to make a reference, a better reference, but also for an iconic individual. So this is where we are. This is also ongoing. We've generated long-read data of 2 kinds. We've done a de novo assembly. We've kind of polished that data with PacBio, with the Illumina short-read data. But what we really need to do is scaffolding, or kind of a chromosome-level mapping, and that's where we still are. So basically, this is just to show you some very boring statistics. This is the existing reference assembly. This is another assembly which is part of that range-wide genomics paper I talked about early on. And this is our assembly. But what remains is basically the scaffolding. Okay? So basically, what does that mean in common language? So this is a cat chromosome, just one of them. And this is all the contigs that we have for tiger, all of these, which map to the cat very well. But putting them together, which is next to which, right? That is something which is only possible once we do scaffolding. And it's important because when we infer these long runs of homozygosity, if our reference is not adequate, we may make mistakes. So unfortunately, the scaffolding failed from the Machli sample because we were able to only get to it a day after the animal died. and the cells were no longer intact. So what we're trying now is to very actively, quickly get to the site where an animal is being darted or so on, so we can get intact cells to finish this this assembly. So anyways, that's something which is ongoing, and I should just say that most of the reference genomes that exist are all from zoo animals, right? And those animals have been under different kinds of selection and so on. It's very rare to have a reference from a wild animal. So if we can pull this off, it will really be very nice. Okay, so how much time do I have? Sorry, 5 more minutes? Okay, good. So I'll then just tell you very briefly about this other story, another isolated population. This beautiful animal is a melanistic tiger, and it's a phenotype. This is a camera trap image seen only in Simlipal, not recorded visually through camera traps anywhere else. So this was something actually which we were approached, which is a very exciting and interesting question. Why are these tigers in Simlipal, and what is the genetic basis for this phenotype? Okay, so there's a lot of work on color variants in tigers by a group in China, and they've identified mutations for the white tiger, for the golden tiger, and the snow white tiger. However, what we have in the melanistic tiger is actually a pattern disruption. This is a paper on cats where Kalin et al. Showed that mutations in this gene called TACPEP, which is a transmembrane peptidase, during development changes pattern from this kind of a regular stripe to this kind of whirly pattern. And so we wondered, is it the same gene which is implicated in this melanistic tiger, which seems to be a pattern variant? And so again, luck had it that in the Nandankanan Zoo, there was born a melanistic, well, 2 individuals which are melanistic. So here's the white mother. Here's the daughter, who's white melanistic, independent traits. And here's the son, who's orange melanistic. So now we get this opportunity to take these individuals and their pedigree and try to understand the genetic basis of this trait. So we got lucky. It was really simple. It turned out that basically the mother and the father were carriers, and the melanistic tiger and the other one basically had 2 copies of this mutation. And this mutation basically is a missense mutation, which causes a major change from histidine to tyrosine. And this particular amino acid is within the active site of this protein. So when we actually look at this, this is still association, but when we actually do look at the LOD score, it's 100 times more likely that it's this mutation versus something else in our zoo pedigree. So it's all still in the zoo, right? So maybe it's a different mutation in the wild, very unlikely, but that's often the response we got. So now we went back to the field. Oh yeah, by the way, this particular, the Nandankanan Zoo had 1 individual from Simlipal somewhere back in the past. Okay, so we went to Simlipal, a very different context. 2 months, we did not see a single tiger. Okay, so you can't follow animals here. But we did find a lot of scat, and when we did, identified individuals, scored the mutation, we found that there was both the melanic and carrier individuals which were detected, and basically we found the frequency of this allele was 55%. So that is really high, and that suggests drift. This population is possibly isolated, because so far we've started looking at more individuals, but we've looked at 60 individuals outside Simlipal, and not a single one is even a carrier. So that means that the frequency is at least 1/60 somewhere outside, whereas it's 1/2, right, in Simlipal. So what has enabled this dramatic change in the frequency? Possibly isolation. So the first question I said is, why are there melanistic tigers there? I can't answer that, but there could also be because there's only about 15 tigers here, and it's 2 family groups which are inbred, right? So this is something which we need to find out more about. We've not done that work yet. But just to give you this example that it's not only Ranthambore where we see this. So just to conclude quickly, I think it's really important to understand the effects of inbreeding in wild populations, because whether these populations survive or not in the future will depend on whether we would like to infuse genetic variation from other places, or if there are no fitness effects, potentially allow them to continue as they are. Indian tigers have very high genomic variation, genome-wide variation altogether, but individuals show strong signatures of inbreeding. And this is actually a very interesting result, possibly because our habitats are very small. Think of the Russian Far East, huge area, very few tigers, but they have large areas to move around, and so probably mixing around much more, whereas our tigers are hemmed into much smaller areas. Ranthambore is a good candidate to study these kinds of things, particularly inbreeding, because we can follow individuals and possibly reconstruct pedigrees. Tigers here do seem to be inbred, and inbreeding may be higher in younger individuals. I'm not sure about this yet. Reconstructing the pedigree with hair samples to identify inbred individuals is what we want to keep doing, and then follow the fates of these individuals. Of course, Ranthambore is also, like I said, a heavily managed population. So what we've managed to do now is to get records. So what I mean by that is, when an individual is sick, there's intervention. Okay, so we've managed to get all of those records. So hopefully we can also, if not look directly at fitness, try and understand whether, you know, an individual was managed more, and so on. Sorry, I wasn't very clear about that. Anyway, and basically we are working on this high-quality genome assembly, and isolation does seem to result not just in, you know, when you say, oh, there's low variation, it sounds very abstract, but here we are actually seeing a phenotypic frequency change in a small and isolated population. I'd like to continue working on, of course, I have to work on this for a long time, but I'm really interested in looking at deleterious mutation load, and for example, we can harness inbreeding data on zoo tigers to look at what particular traits show up with inbreeding. For example, crossed eyes is one trait which shows up often in inbred individuals, and so on. And what regions do those map to in the genome, and then look for those regions in our wild tigers. Are there consequences? This is, of course, a very big question, and I am not sure. I often wake up at night wondering, maybe this is all, I mean, there's inbreeding, but maybe it doesn't matter. I guess we'll have to find out. And what's the long-term strategy to avoid this situation? Okay, none of this is possible without, of course, support. There's no way I could have done this work. I mean, this work requires a lot of support of various kinds, not just financial, but logistic and a lot of hard work. And I have some great students. There's a field team in Ranthambore. Mujahid here, who's been working on tigers for the last 10 years. He knows more about them than we probably will ever learn. And of course, my students. And thanks all for listening.
That was really fascinating. The question I had is, what is the trait that you're going to look for to measure fitness? Yes, in terms of inbreeding. What's the outcome that you're going to judge to see whether fitness is compromised or not?
So I think that basically the best strategy is to follow this population into the future and follow it from a perspective of reproductive success. I think in terms of the traits itself, it might be more fruitful to look at inbred individuals where you know, in zoos, where you know what the inbreeding coefficient is, and to see whether the same stretches of homozygosity are, you know, is there a pattern for inbreeding? Is there a structure in the genome for it? It's unlikely, but so far we do see common stretches of, between the Ranthambore tigers and other tigers as well, the same region is homozygous. So, I mean, I might be wrong, but I think that would be
so reproductive fitness is going to be your measure of consequences of inbreeding.
Yeah. So, but that'll take a long time, no?
Yeah, right.
Yeah.
Uma, really nice presentation.
Thank you.
So I have a conceptual question and a follow-up question on that. The conceptual question is, in terms of looking at epigenetic imprinting, in terms of differences between inbred and less inbred and more outbred tigers.
Yeah.
Have you looked at, for example, in your genome sequencing or methyl sequencing, the methylome differences between those? And I'll also ask you the follow-up question so you can answer them both. The follow-up question is, since you're doing so much work with these, have you thought of isolating something like fibroblast cells or even from blood, transforming them into induced pluripotent stem cells, so you can then look at the consequences to development at various levels, I mean, between less inbred and more outbred tigers?
Yeah. So second question first. I've thought about that a lot. Basically, we need to, and we've been working towards that. So let's see if we're able to do it. The main thing is I would really like to do it from a tiger from the wild, but we may have to just end up doing it with a zoo individual. So the first question, so I think we know so little about non-model species that I think it might be a little hard to make inferences, conclusive inference. Again, they would be like the ROH kind of correlative.
Right, but the simple question is, does increased homozygosity in stretch of sequences imply reduced epigenetic changes between the parent and the progeny, or is it going to show show a random effect in terms of not being relevant?
Yeah, so I haven't looked at that yet, but that's something definitely which we could look at. Sounds like a good idea.
Yeah, so while I see few more hands here, but in order to be on time schedule, I think we'll have to close the session now. Uma is available during the lunch break to discuss if you want to. Thank you very much for your attention, and thank you, Uma, for a very good talk. So we'll break for lunch now. We will meet at 2 p.m.